A very good afternoon to all the biology students out there. For this edition of the biology quarantine tutorial videos, we will be looking at plant nutrition. Okay, so I've specifically chosen an experiment because it's as Namibian students always struggle with the paper 3 questions. Okay, so in plant nutrition, there is one of the most important experiments that students need to know, which is testing if a certain factor, which might be carbon dioxide like a chlorophyll, is necessary for the process of photosynthesis. Okay, so when we look at this experiment, students need to know that we need a control of the experiment. Okay, so what is a control? Okay, so a control is basically a sample that is given all required characteristics or factors. Okay, so for example, for a leaf to carry out photosynthesis, it needs light, chlorophyll, and carbon dioxide. Okay, so when you are asked to carry out the experiment like this, and then you are asked what is the control of this investigation, you mention that it is the leaf which was exposed to all factors. Okay, so when you carry out this experiment, also notice that to make the experiment fair, you need to discharge the leaves. So what is the reason behind discharging a leaf? Okay, the reason behind discharging a leaf is the you want to test if the leaf will photosynthesize. Okay, so if the leaf already contains starch, then it won't make your experiment fair. So you need to remove the starch. So how do you remove starch from a leaf? So the most common way of removing starch or discharging a leaf is by placing the potted plant in a dark cupboard at least for 48 hours. Okay, so when you place it in a dark cupboard for 48 hours, you prevent it from getting sunlight. Okay, so by doing this, by so doing, you prevent it photosynthesizing and thus starch is not formed. Okay, so after you have discharged your leaf, you now want to test, or you now want to carry out the whole process of photosynthesis. Okay, remember we have two potted plants. One of the potted plants must be placed in all required factors of photosynthesis, which is chlorophyll, um, sunlight, and also carbon dioxide. But the other one must be placed in such an environment that one of the factors is blocked out. Okay, so for example, let's say we place one of the potted plants outside a classroom and then the other one we keep it inside the cupboard. So when we keep it inside the cupboard, we know that this plant is not exposed to sunlight, while the other one outside the class is exposed to sunlight. Okay, so we wait at least for about 24 hours and then we collect now these potted plants from their environments and then we bring them to the laboratory. So when you bring them to the laboratory, we remove a leaf from each potted plant, okay, from the one that was exposed to all factors and the other one that was placed in the cupboard. Okay, so before we test this leaf for starch, we have to boil the leaf in water at least for 30 seconds. Okay, so why do we do this? Why do we boil the leaf in water for 30 seconds? This will kill the cells in the leaf and break down the membrane. By doing this, it allows iodine to penetrate the cells and then it will react with the starch, creating a blue-black color. After boiling in hot water, we also need to boil the leaves in alcohol. Okay, we need to boil the leaf in alcohol. The green leaf of a leaf and brown iodine solution can look blue black together, so you need to remove the chlorophyll by dissolving it with alcohol. Okay, so what is the next step? We rinse our leaf in water so that we can soften it. Okay, because alcohol, when it reacts with leaves, it kind of make the leaves to be brighter. Okay, and when something is brighter, it can easily break. We don't want to break the leaf. Okay, so we place it in water for us to soften it. Okay, so after you have listened, then you place your iodine on top of the leaf, and then you get your results. Okay, so if starch is present in a leaf, then the leaf will turn blue-black when iodine is added to it. But when it did not photosynthesize, the plant will simply remain brown. Okay, thank you. I hope this was really useful, and stay safe.